everyone. In this video, we'll continue our description of the events that occur in the renal tubule, particularly reabsorption and secretion, by describing what happens specifically at the distal convoluted tubule and collecting duct. So in order to orient ourselves to the picture, we notice that the orange cells make up the DCT and then the pink cells make up the collecting duct. All the cells are simple cuboidal epithelial cells, but I just want to mention in the collecting duct the presence of two particular cell types, and those are intercalated cells and principal cells. And physiologically, we can notice the difference right away between them is that the intercalated cells actually have abundant microvilli, whereas the principal cells do not. Also, in comparing the two cell types, the intercalated cells have abundant mitochondria. They are actually responsible for maintaining acid-base balance in this section of the renal tubule, and there are actually multiple subtypes of this cell. Let's just get started by describing some of the transport mechanisms. So as before, this is not comprehensive to all of the transport mechanisms. It's just to sort of offer an overview of some of the things that are happening in this particular section and how they may be different from the other sections. So we notice in the distal convoluted tubule, we have the opportunity to reabsorb sodium and chloride. And we actually have a co-transport mechanism for sodium and chloride at the apical surface. And then that's connected to an active transport mechanism for sodium, which is, of course, the sodium-potassium pump in the basolateral surface, and a chloride ion channel. So we know that the presence of the sodium-potassium pump is what maintains the gradient for sodium and actually allows the co-transport of sodium and chloride to be secondary active transport. And then similarly, as the chloride accumulates inside of the cell, we maintain the gradient for the chloride to move down through the ion channel into the ECF. Next, let's talk about potassium secretion. So we've talked only briefly about potassium, especially in the sense that in the PCT, we're able to reabsorb some potassium through paracellular transport, but we really haven't described the secretion of potassium yet. And it's very important to note that regulation of potassium in the plasma is extremely critical to so many bodily functions, and electrolyte imbalance that involves hyper or hypokalemia can become very severe. So there are certain opportunities for secretion of potassium as needed. So in this example, we'll notice a sodium-potassium pump that's present in the basolateral surface of the principal cell, and that we of course expect to be there anyway, but the potassium moves into the cell where it's already highly concentrated, and then we establish a potassium ion channel in the apical surface where potassium is simply able to move down its concentration gradient into the renal tubule. So as we need to secrete more potassium, we're able to allow the potassium to simply move down the ion channel. Okay, next let's describe management of acid-base balance in the kidneys. So we know, of course, that acid-base balance can be maintained by the carbonic acid bicarbonate buffer system in the plasma, and it's possible for that process to work in a very rapid fashion, but over a longer term we actually can use the kidneys as well to maintain acid-base balance. So bicarbonate ions are actually filtered at the glomerulus, so we do want to reabsorb any bicarb ions that are filtered, and that actually begins in the proximal convoluted tubule, but any additional management of acid-base balance is going to occur by selectively secreting or reabsorbing hydrogen ions or bicarb ions as needed. Let's start with the idea that we need to secrete more potassium and reabsorb more bicarb ions. So the cells, the intercalated cells actually possess particular enzymes that are able to catalyze the conversion of CO2 and water to bicarbonate ions and hydrogen ions. So the same family as carbonic anhydrase that we know in the red blood cells, we're actually able to use the enzymes to catalyze the conversion. So the renal epithelial cells are able to produce bicarb and hydrogen ions as needed. So if we actually need to reabsorb more bicarb ions so that we can manage the plasma, if the plasma pH is too low, for example, and we need to combine the hydrogen ions with the bicarb ions to make CO2 and water, we will produce the bicarb and the hydrogen ions inside of the intercalated cell, and then we can take a look at some of the transport mechanisms that are available for both. So in the apical surface, there are two options for secretion of hydrogen ions. One is an ATP-powered pump that exchanges a hydrogen ion for a potassium ion, and the second is an ATP-powered pump that simply moves the hydrogen ions into the renal tubule. And then in the basolateral surface, we'll have exchange of bicarb ions for chloride ions. So quite simply, negative charge for a negative charge. The bicarb has accumulated inside of the cell, so it's moving down its gradient into the interstitial space. And because it's exchanged for a chloride ion, the chloride ion actually is able to just move back into the interstitial space afterward. Now, conversely, if we needed to secrete more bicarb, so if our pH was too high, we need to reabsorb more hydrogen ions, we can actually use a different type of intercalated cells to sort of do the opposite function. In this case, we also know that the hydrogen ions and bicarb ions are being produced inside of the cell, and then at the apical surface we'll have secretion of bicarbonate ions in exchange for chloride ions, so again negative charge for a negative charge. And in the basolateral surface we'll have a chloride ion channel for passive reabsorption of chloride, as well as an ATP powered hydrogen ion pump. Alright, so now let's just talk a little bit about the osmolarity of the tubular fluid. So we know that as we return from the loop of Henle, we actually have a very low osmolarity, meaning that there's a particularly high concentration of water relative to the amount of salt, or a particularly low concentration of salt, however is easier to think of it. And so it really is in the DCT and the collecting duct where we're really going to find the most regulation in terms of how dilute or concentrated the urine is going to be. So we know that water is reabsorbed through aquaporins, and aquaporins are always present in the basolateral surface of the DCT and the collecting duct. But the expression of aquaporins in the apical surface is really based upon secretion of antidiuretic hormones. We're actually going to do the mechanism for ADH in a couple of moments, but I just want to point out that the water gradient exists here, so it'd be very easy for the water to flow from the renal tubule back towards the peritubular capillary through the renal epithelial cells, but we have to have expression of aquaporins at both surfaces, and the one that we particularly regulate is the one at the apical surface. As I mentioned, we'll do the mechanism for ADH, and we'll also do the mechanism for aldosterone, but we're not going to do the mechanism for parathyroid hormone. So I just want to describe quickly the reabsorption of calcium that occurs in the DCT as well. So we already know that calcium can be reabsorbed in the proximal convoluted tubule, and additional calcium reabsorption can occur in the DCT. When thinking about the hormones, particularly parathyroid hormone and calcitonin, one of the means to regulate plasma calcium concentration is to utilize the hormones to increase or decrease the reabsorption of calcium in the renal tubule. So now we're just going to sort of talk about that for a moment. So we notice that there is a calcium ion channel that's present in the apical surface of the cells of the DCT, and then there are actually two different options for transport of calcium across the basolateral surface. The first is familiar NCX, the sodium calcium exchanger, so exchange of calcium for sodium ions, and that's counter transport or antiporter, but it's not ATP powered. It's just would be more along the lines of secondary active transport or facilitated diffusion. And then we also have a specific calcium ATP pump that pumps the calcium into the interstitial space. And so with the presence of parathyroid hormone, we can actually increase the expression of these different transport mechanisms for calcium, thereby increasing calcium reabsorption in the DCT. All right, so of course we mentioned that the transport mechanisms discussed are not comprehensive. There are still other possibilities, but those are just the ones I want to highlight for this particular section. Now let's take a look at the mechanism for antidiuretic hormone. So we know, of course, the hypothalamus contains a cell bodies for some neurons that are responsible for production of antidiuretic hormone, and the axons for these neurons travel from the hypothalamus to the posterior pituitary, where we can synapse with blood vessels and release the antidiuretic hormone into circulation. So the first question is, how does the hypothalamus know that there should be an action potential to release this ADH? And that actually is based upon osmolar receptors that are present in the hypothalamus. So a high concentration of solute in the plasma will trigger the osmolar receptors to release antidiuretic hormone. And so basically the plasma obviously is going to circulate through the hypothalamus, and then these receptors can just recognize this concentration. And so then we'll get an action potential from the hypothalamic neurons, and it will release ADH at the posterior pituitary into the bloodstream. So antidiuretic hormone travels in the blood to renal epithelial cells. So we'll just actually use an example of a cell of the collecting duct at first. And on the basolateral surface of the collecting duct cell, we have a V2 receptor. So we know that antidiuretic hormone is water soluble, which means it's going to work by second messenger. And so the antidiuretic hormone actually binds to the V2 receptor. And I just want to mention, we know already that antidiuretic hormone and vasopressin, those terms are one and the same. So actually, when we administer vasopressin through a peripheral IV, the vasopressin actually binds to V1 receptors. So this is a different version of the receptor, but it's still able to bind ADH. This V2 receptor is expressed particularly in the renal epithelial cell as compared to the V1 receptors that are located peripherally. So we bind the antidiuretic hormone to the V2 receptor. This particular receptor is associated with G protein, so it's a G protein coupled receptor, which means that we're going to have a cascade of reactions that ultimately results in the generation of a second messenger. In this case, the second messenger is cyclic AMP. Now, we mentioned previously that the aquaporins are always present at the basolateral surface of the epithelial cells, but we can decide when the aquaporins are present at the apical surface. So the aquaporins actually can be synthesized by the renal epithelial cell, and they can be stored in vesicles until they're needed. And then upon the arrival of the second messenger, based on this cascade of reactions, we can actually move that aquaporin to the apical surface of the cell. And of course, once we have aquaporins at both surfaces, we know that the osmolarity of the tubular fluid is very low, and the water is in very high concentration, and the water will diffuse passively down the concentration gradient. So it's being reabsorbed initially across into the epithelial cell, and then into the interstitial space, and then into the vasculature, the peritubular capillary, where it can be retained for the body. Now, when the plasma osmolarity is low, the antidiuretic hormone will be inhibited, and when the antidiuretic hormone is inhibited, the aquaporins can actually be removed from the apical surface and moved back to being retained in vesicles inside the cell until they're needed at a later time. All right, and then lastly, let's consider the mechanism for aldosterone. So thinking back to the endocrine system, we know that in the hypothalamus we have neurons that synthesize and release CRH, or corticotropin-releasing hormone. This hormone is released into the hypophyseal portal system and transferred to the anterior pituitary, where we will have subsequent synthesis and release of ACTH, or adrenocorticotropic hormone, which has the ability to act on the adrenal cortex in times of stress. And we also know, of course, that even sort of more than the ACTH, increased plasma potassium or release of angiotensin II can also trigger the release of aldosterone. So aldosterone is released into the plasma and then transferred throughout the body, but most importantly to the cells of the distal convoluted tubule and the collecting duct. So we'll just draw on some cells and take notice of the basolateral and apical surfaces. And like any other cell, there's nucleus, endoplasmic reticulum, Golgi apparatus, and so on. As the aldosterone arrives to the cells of the DCT and the collecting duct. We know that aldosterone is a steroid hormone, so it's able to diffuse right through the membrane. And actually, when we described cortisol, we said that particularly we have cortisol binds to a glucocorticoid receptor, and then there's a cascade of reactions that allow for that complex to move into the nucleus and interact with the DNA. So the same thing is going to happen here. We're actually going to have aldosterone binds to a particular receptor that's waiting for it inside of the cytoplasm, and then cascaded reactions will occur and will transfer into the nucleus where we can communicate with the DNA. So what we're going to get is upregulation of the sodium-potassium pump in the basolateral surface and increase in sodium channels in the apical surface. So essentially what we're doing is synthesizing more channels for sodium and then more sodium-potassium pumps, which means that we can increase sodium reabsorption in the distal convoluted tubule and the collecting duct. And we know that when we have increased sodium reabsorption, we're now simply increasing the gradient for water reabsorption, which means more water reabsorption, which means more water present in the peritubular capillaries being saved for the body, which means increase in blood pressure and blood volume. And we also know that we initially released aldosterone because of an expected decrease in blood pressure or blood volume, so we're simply correcting the problem. Now, one other thing that's noticeable about aldosterone is that we actually get an increase in the production and arrival of potassium channels in the apical surface as well, which doesn't have a significant acute effect, but over the longer term, so if we have chronic release of aldosterone, sort of like chronic stress that leads to chronic release of aldosterone, we can actually get increased potassium secretion and excretion. And one thing that's interesting about this is that one of the triggers for a release of aldosterone is increase in plasma potassium. So if we need to excrete some additional potassium, if this is a chronic issue, this is sort of an additional opportunity to do so. All right, so that actually concludes our description of the events that occur in the renal tubule. We finally made it to the end. And basically everything that's left in the collecting duct at this point, after all of the reabsorption and secretion, is what's going to be excreted. So we'll eventually dump into the renal pelvis and then ureters and so on. So in our next renal video, we'll take into consideration what happens in the rest of the urinary system. But that's all for now. Hope you enjoyed it. Thanks for watching.